Thank you all for coming today. This is Grand Rounds talk that's hosted by the Division of Biostatistics in the newly named Department of Public Health Sciences. My name, for those of you who don't know me, I'm Sunil Rao. I'm the Director of the Division of Biostatistics. So again, welcome. Today you're in for a real treat. I mean, when we were choosing our guest speaker for this Grand Rounds talk, we wanted somebody that was really at the bleeding edge of genomics research. And that sort of comes from a variety of different angles. Broad appeal in the sense that genomics is kind of innovating everywhere in the basic sciences, the medical sciences. We're all facing it in one way, shape, or form. It also dovetails really nicely with a lot of our in-house groups that are doing their own work in genomic analysis, mainly from our division, The Hague, the Center for Computational Sciences, and so on. Lastly, we wanted somebody who's a great storyteller, and John is a great storyteller, you'll see. So let me give you a little bit of background about Professor Quackenbush here. He's a professor of biostatistics and computational biology, as well as cancer biology at the Dana-Farber Cancer Institute. He's the director of the Center for Cancer Computational Biology at the Dana-Farber. He's a professor of computational biology and bioinformatics at the Harvard School of Public Health and the Department of Biostatistics. Interesting background. We talked about how he got into this field. So he comes at it from a little bit unusual place. He's got a bachelor's in physics from Caltech. Then he did a PhD in theoretical physics from UCLA. Where's the bio part? You know, he masked it. He then did a postdoc in physics. He then got a career award from the what's now the National Genome Research Institute, which got him into the bio side of things. Spent 2 years at the Salk Institute after that, working on physical maps of human chromosome 11. 2 years at Stanford after that, developing new lab and computational tools for sequencing the human genome. In 1997, he joined TIGER, the Institute of Genomic Research, and he focused on microarray data analysis and developed new analytical tools for integrating data across domains. And in 2005, he moved to Harvard and the Farber. His current research is on the analysis of human cancer using sort of a systems biology-based approach to understand and model biological networks. His group's made fundamental discoveries about the role that variation in gene expression plays in defining biological phenotypes. He's got a nice book in 2011 called The Human Genome, Book of Essential Knowledge. He's the editor-in-chief of the journal called Genomics. He's a member of many, many national committees and advisory boards, and he's given many named lectures, including at Purdue, University of Bern in Switzerland, and the Ontario Cancer Institute in Toronto. So it's a real pleasure to have him here, and I'd like to give him a warm welcome.
I always hate being introduced because it always makes me feel like I have something to live up to. But it's always an honor to be introduced by someone whose work you also truly admire. So thank you very much for the invitation and the kind introduction. When Sunil asked me to come here, he asked me what I would talk about. And I said, well, I sort of have two sort of general talks that I give. I talk a lot about the infrastructure we have and put in place to build personalized medicine. And also, I have my own research interest, and they're sort of focused on trying to understand biological systems and how we put them together. And I think he said, well, can you do both? And so I'm going to try to do both. I'm going to tell you a little bit of a story. But you can't do one. You can't do the research without putting the systems in place. And this is something that I think we have been wrestling with for a long time, and something from talking to people, people here all day today, really recognize that you, like everyone else, is wrestling with it. And this is the problem of really thinking about genomic data and how we do personalized medicine. So how do we get from where we are to where we want to be? The genome was sequenced in 2000, 2001, 2005, whenever you declared finished. And the promise was we were going to change healthcare and change biomedical research. But we haven't quite gotten there. But we have the opportunity to do it. And so I'm going to start by telling that story, but then I'm going to tell you a little bit about the research we do. So I like to use quotes in my talks, and I'm actually going to do two things here. The first, I'm going to talk about this problem of how we do personalized medicine. And as Sunil told you, my background's in physics, and so what better way to illustrate the importance of biology than use a quote from a physicist, Jules Poincaré. Poincaré said, science is built with facts as a house is with stones, but a collection of facts is no more a science than a heap of stones is a house. And this is a quote I've been using for years to talk about genomics, because what genomics increasingly is giving us is big piles of data. But the intellectual challenge we face is to take that pile and turn it into something useful. To take data and to turn it into knowledge, take that knowledge and to turn it into understanding. understanding. So that's one of the intellectual challenges that I think we really face. The other piece, which is sort of my research work and passion, is in trying to build models of biological systems. And this is a quote from Sam Carlin, a biomathematician who's at Stanford. Carlin said, "The purpose of models is not to fit the data, but to sharpen the questions." And you'll see that philosophy emerge as I start to talk about how we think about building network models and what we do to get there. So, what's driving all of this? Well, for me, it's really the idea that these big data sets that we generate, these genomic data sets, have allowed us to think very differently about the course and progression of human disease and how we actually understand it and how we better deal with it and try to treat it. So by having a lot of genomic data, we can start to do things like think about predicting people's genetic risk. And when the first genome was sequenced, everybody said, we're going to find all the common variants and it's going to tell us about common disease, and now we recognize the problem is much more complex than a handful of risk alleles. That really, disease is very complex. Nevertheless, we can look at genetic backgrounds and try to do better prediction of risk. Once we do that, we'd like to be able to do better early detection. We'd like to be able to identify who has disease. And what's really exciting now is that genomic technologies are starting to allow us to do that in ways that we couldn't have imagined, looking at circulating tumor cells or looking for circulating DNA. But beyond just this kind of analysis, we're also looking at new interesting imaging modalities and approaches to imaging analysis, and really thinking more and more about how we can find diseases early, because the earlier we detect them, the better we can usually treat them. But once we have a patient with a disease, we'd like to stratify them. We'd like to understand what molecular characteristics are active in their disease that distinguishes them from other patients that have histologically very similar, very similar diseases. Because we understand that some patients respond to therapy and some don't, which means that there must be some molecular mechanism that's different in its operation between those patients. So we want to stratify patients, we want to stage disease using molecular information, but ultimately we want to use all of this to try to predict the right treatment option to improve outcomes for our patients and improve their quality of life, right? We're all going to die, we just want to be as far away and as pleasant as possible. And that's true of our patients too, right? We want to give them the best quality of life for as long as possible. And we want to give them the data to allow them to make informed decisions about what's best for them. And so a lot of the work we've been doing over the years has been driven by access to large data sets, largely microarray data sets, whether it's gene expression data sets or genome-wide association SNP genotyping data sets or copy number variation data sets. A lot of them come from arrays. But what's really changed everything in a very profound way is DNA sequence data. And these new sequencing technologies that are not that new, they're about 6 or 7 years old, but they're really starting to drive the generation of large, complex, integrated, multi-omic data sets. And it's changing the way we think about a whole host of different problems. So how do you turn this vision into reality? Well, the first thing you need to do is have access to samples and rational consent. And I'm going to talk about each one of these issues. You need to develop some kind of technology platform. You have to start generating data. You have to think about information integration. And for me, this is part of my work on a day-to-day basis. How do we bring information together so we can actually do things with it? And I can give you some examples of that. I have lots more examples if you want to talk about it. You have to present data and information back to the local community, right? You have to take what you're doing and communicate it to other people and give them tools so that they can do it, realizing that the number of quantitative geeks like Neil and me and some of the other people I talked to today are limited in number, but lots of people want to have access to this data. And you really want to, in many ways, democratize access to the data and information. You want to enable research beyond your own. I heard an interview with Penn and Teller, and Penn Jillette said, he really admired Blue Man Group because what they were able to do is take 3 guys, develop an act, and clone themselves, right, by painting themselves blue. If I could only paint myself blue, I could, and paint other people blue, I could help a lot more people analyze their data. So how do you actually do that? How do you enable research beyond your own? This is something we're increasingly recognizing we can't do ourselves, and we're trying to strategically engage corporate partners. You have to conduct research, though, to really keep all of this moving forward. What we know how to do today with the data sets we have is not going to touch what we can do and should do with the data sets we have tomorrow. And then the last thing is that we have to communicate our mission to the broader community, in large part to help drive this entire process. We have to educate patients. And so I'm going to touch on all of these more or less in this order. The first topic is really ensuring access to samples and rational consent. And in doing research, this is something you have to really think about carefully. We have a personalized medicine program now at Dana-Farber. It started a little bit more than a year ago, I guess about 15 months ago. We spent probably 6 months talking about how we were going to implement the assay, and more than 12 months talking about how we were actually going to structure the consent to get patients involved in the process. And this is really a huge issue, but it's driven by the fact that patients want to be part of the process of curing disease. They really do. They want to consent, they want to contribute to studies. However, we have to recognize that we have to structure the informed consent in ways that allow them to do that, yet communicate what the subtle risks are associated with using genomic data. HIPAA requires that we provide informed consent, but also that we assure patient confidentiality. And this is one of the big challenges, because genomic data is fundamentally different than any type of data we've wrestled with before. There have now been 2 or 3 studies published where people have taken genome data and publicly available information like driving license records and other public data sets and traced genomes back to individuals. Right? If you have about 250,000 base pairs of your DNA, there are enough unique variant positions in that 250,000 base pairs, right? And that's one tiny piece out of 3 billion bases, or 6 billion if you have the diploid genome. 250,000 bases have enough variant positions to meet the FBI's definition of forensic identifiability. Okay? A tiny snippet of your DNA is enough to find you. So we have to really think about that, because identifiability has become a moving target. So, we have a lot of structures for storing identifiable information and data, but they don't often scale well with storing genomic data. So we really have to think carefully about that. We have to think carefully about identifiability, and we have to think carefully about how we inform patients of how we're going to use their data and what we're going to use it for. But with the $1,000 genome here or coming soon, in the age of Facebook, what this means is unclear. Younger generations, and this is really a generational question, because younger generations have a very different view of privacy than older generations do. Okay? My nephew is 20 years old. He's on Facebook. His mother is his Facebook friend. And she sees things and comments on things that he's doing today that I won't tell my mother I did when I was back in college. Very different view of how we deal with data and privacy. People click through licenses all the time. But we have to think carefully about what this means, because this data can be used and misused. And so we really have to think not only about the identification, which always doesn't work, but really the overall process of data encryption and data security and how we manage it. So there are a whole host of issues you just have to be cognizant of. But what's really emerged for us is the idea that genomics has been a disruptive technology, and we have to rethink the way in which we manage this data. Okay? I met my wife on Match.com. I guarantee my son is going to meet his wife. He's 7. He's going to meet his wife on GeneMatch.com. Okay? But by the time he's 15, his genome is going to be sequenced. I almost guarantee that in the next 8 years. I just don't want his genome on Facebook. Right? So we've got to think very carefully about this entire process and realize that the technology is rapidly outstripping our ability to come to grips with the data that we're generating. There are all kinds of rules about data generation, but if I can put a Q-tip in my mouth and mail it to Beijing and get my genome back for $1,000, all the rules and regulations aren't going to matter. So we really need to think very carefully about what we do with this kind of technology. At some point, though, we have to develop a technology platform. If you're going to do personalized medicine, you have to start generating data. And really, my thinking about this has been driven by this cost curve that's available from NHGRI. Actually didn't like the graph they had on their website, so I downloaded the data and started adding my own to it and plotted it. But this is the graph of the cost of sequencing genomes starting when the first genome was declared finished in 2001, when if you were going to start another project, it would have been about $100 million. And then it dropped over time. And so if you're a quantitative geek like me, you want to fit a regression line to it, you realize that doesn't make sense, because you don't have one data set here, you actually have 2. You have the old Sanger sequencing data set, and then you have this second generation of sequencing technologies. And the amazing thing was there was a long period of time here where the cost of sequencing genomes was dropping consistently by 33% per quarter. Every 3 months it was dropping by a factor of a third, okay? Which is just astonishing. People always say it's faster than Moore's Law, which doesn't make sense because Moore's Law is about the doubling of the number of transistors on a chip every 18 months. But basically you're doubling the amount of information much faster than every 18 months. And so that you can get from a single asset, or per dollar, I guess. But the way I really think about this is that, if I look at this and sort of project forward, I would have predicted, and I did this a few years ago, we hit the $1,000 genome in October of 2012. I'm still hoping we get there. But the truth is, sequencing a genome today is about $3,000, okay? And that's a statement that changes the way we think about generating data. Because over here, right, even a few years ago, when it was $100,000, I used to tell people in all seriousness that if I had cancer, I would mortgage my house and sequence my cancer genome, okay? Today, I can put it on my credit card. And that changes the way we think about personalized medicine, it changes the way we think about biomedical research. If you extend this further, you hit the $100 genome sometime in February of 2014. Are we going to get there? The answer is no, not in 2014. But I would firmly predict that we're down near or below $1,000 by next year. And this trend is just going to continue. There are a whole host of technologies beyond the second generation of sequencing instruments that are promising to deliver $100 genomes or $10 genomes in very short periods of time. So, this is actually, I maintain a wet lab and we do sequencing, and this is part of this is one of the instruments we have. This is the Illumina HiSeq. And we can do basically a genome in about 7 days for on the order of $3,000. And again, it's really changed the way we think about generating not only whole genome data, but gene expression data, epigenomic data, data all over the place. This instrument actually got me very excited last year. In January of 2012, Life Technologies announced the ion Torrent Proton, and their promise was it was going to deliver a $1,000 genome in about 24 hours by late last year. They never quite got there, but it put a lot of pressure on the whole field to drive technology forward. And there are other players that are building instruments. This is the Oxford Nanopore MiniON. This was announced at a meeting in Marco Island last year. It's the USB sequencer. It actually uses DNA nanopores. And what it does is it detects as this is a DNase, but really the way they implement it is with the DNA helicase that feeds the DNA through this nanopore. And you measure micro differences in the current flowing through the nanopore because the different bases have different physical sizes. And so their claim, which remains to be seen, is that they could build an instrument that would sequence a genome in 15 minutes with a disposable sequencer that would cost less than $100. Okay? So are we there today? No, but we're definitely moving in that direction. So the challenge now is these new technologies are really transforming biomedical research. It used to always be thought of as a laboratory science or clinical science, but increasingly I think we're coming to recognize that a big part of biomedical research is understanding that it's an information science. We need new approaches to make sense of all of the data that we generate, but the winners and losers in the race to understand disease are ultimately going to be those who best collect, manage, analyze, and interpret the data they can generate. Okay? So that's been a driving principle behind a lot of the work that I and the group have been doing. How do we collect all of this data, put it in one place, and then once we do that, how do we actually learn something from the underlying data? So we focused over the last few years on making information management and information integration a central mission. And really, I arrived at Dana-Farber in March of 2005. In fact, I can tell you my first day is March 14th, 2005. Everybody says, oh, do you hate being there? You count from the day. And I say, no, first, if you're a nerd, you realize March 14th is Pi Day, okay? Secondly, my son was born 1 year later on March 14th, so I always have a reminder. Third, it's Einstein's birthday. So if you're a physicist, you have to remember. So I have all these things against me. But I arrived there in March, and within a few months of arriving in March of 2005, I was approached by a group working on a very simple problem. And I've come, even talking to people today, to realize that this is a universal problem. There was a group that was running a clinical trial, and they had a database in which they had all their clinical trials information. They also had the EHR and the hospital information system, and they were generating genomic data on these patients in the trial. And in fact, one of the things they recognized was that the clinical data and the genomic data were completely separate. They couldn't integrate it together. In fact, to do it, they were going to different data managers and submitting paper forms and getting, I put somebody to sleep, submitting paper forms and getting these data back in Excel spreadsheets and mixing and matching them by hand. And this is an ultimate recipe for disaster. It's also crazy in an information age. And what I said is, look, we have all these data stores that won't talk to each other. Let's just extract the relevant information we need and put it into a central warehouse. If we can do that, what we should be able to do then is to reach into the data and bring in information from the public domain and really think about taking the new studies we want to do, new clinical trials, looking at chemical biology, whatever we want to do, and synthesize it in a way that we can try to strive towards the goals of improving diagnostics, getting better therapies, etc. So we've been building different resources. As we started to put this together, one of the things we recognized was there's a lot of data in the public domain. One of the questions was, how do we actually get that? So Aideen Kalhan, who worked with me, has been working with me, she had this idea of trying to get one of the data sets that we were very interested in having, but which wasn't readily available, and that was gene signatures. Someone does an experiment, they compare two different disease subtypes, or disease and control, and they use something like a DNA microarray, and they get a list of genes that they say distinguish these. All of the primary data is available in databases like Geo or ArrayExpress, but the results from the analysis were typically in places like Supplemental Table 3 or Supplemental Figure 5, as a JPEG and as a PDF or a JPEG in some online resource, and nobody could easily get access to it. So with Aideen, we developed a very sophisticated text mining approach that I'd be happy to share with you. It's called High School Students. And what we've been doing over the years is paying students to actually come in and transcribe lists and generate these annotation resources. And so now we have close to 5,000 published signatures spanning cancer, pulmonary disease, viral diseases, that we've been using to analyze a lot of different data sets and look for commonalities. But the real thrust of what we were trying to do is to link this kind of data to clinical data. And so we built a warehouse. We actually got a $1 million grant from Oracle to try to build it. We built our warehouse around Oracle's technologies. We then later realized that Oracle's technologies weren't going to help store all the big genomic data. So I want to thank the people from Oracle for funding a project that led us to a different solution. Nevertheless, one of the things we ended up doing was we ended up working with a company called InforSense that then got bought by a company called IDBS. And we built this little portal. And the first application, this is actually the earliest application we have. And if my life is treating me badly, the video's not going to play. I didn't want to transfer my talk on a USB drive because I didn't want this to happen. And if you really want to see it, I'll show it to you. But what this is a very simple, easy-to-use interface. What the interface does is allow you to come in and to look at the clinical data and to focus on the major question we realized people wanted to ask. They wanted to take clinical data, they wanted to take genomic data, and they wanted to take those two independent data streams and use them in the same data resource to construct cohorts. Because fundamentally, almost every question we ask that uses clinical samples starts with a definition of a cohort. I want patients with this disease, with these demographic characteristics, with these molecular characteristics, who did or didn't respond to therapy. And by taking those different pieces of information and putting them in the same data store, what we're able to do is give people a very simple tool for going in without having to understand how to program and to construct cohorts and to ask whether or not samples were available. And honestly, that was the use case that came when Ken Anderson and Nikhil Munshi approached me about this problem of linking clinical and genomic data in multiple myeloma studies. They wanted to be able to understand, first off, whether or not they had enough patients in different groups to even power a study. And we turned that problem into something that would take months of going back and forth with data managers to minutes with them sitting down, being able to look at the data and just ask questions like how many patients meet these characteristics in these different data sets. Once we did that, we were able to link it to genomic data, and I can show you examples of how we want to do that. If people want to ask questions, I can find the video in 30 seconds. So I apologize the video didn't run. But that's the kind of problem that really drove us to think about why this data integration challenge was so important, but one that we really felt that we could address. Obviously, this is part of this broader question of returning data and information back to the local community. And so what I'm going to show you now is actually a project that was funded, not any project we were doing at Dana-Farber, but funded as part of a big stimulus grant that we got in 2009. And this is part of a project that was called the Lung Genomics Research Consortium, or LGRC. It was funded by the NHLBI. And in fact, I can tell you that even though I work in a cancer institute, most of my work that's really cutting edge these days is being done in pulmonary disease. And one of the things about pulmonary disease that's so exciting is NHLBI has made a commitment to funding a lot of genomic studies. And with these new technologies, we can actually generate multiple independent omic data types from the same biological samples from the same patients, because we require smaller volumes of these of sample with these new technologies. And so we were funded to be part of this consortium. And it was a very interesting consortium. In this consortium, what we had were data sets that we were generating based on this NHLBI-funded project called the Lung Tissue Research Consortium that was centered at the Mayo Clinic, where they were collecting lung tissue samples from a variety of different sites and collecting all of their clinical data. What we did was we downloaded all of their clinical data and we loaded it into that same kind of portal that you saw in the non-functional video. And we were able to go in then and look at the patients and construct cohorts based on the clinical characteristics. We selected cohorts that had COPD, chronic obstructive pulmonary disease, and ILD, interstitial lung disease, and controls. We got the samples from the Mayo. They went to the University of Pittsburgh Medical Center where DNA and RNA were extracted. Those were sent to 5 sites around the country where we generated whole genome SNP profiles, whole genome gene expression profiles, whole genome RNA-seq profiles, small RNA-seq profiles, whole genome methylation profiles, and then for a limited number of patients, whole exome sequence data. All of that data flowed back to us. We actually built a distributed LIMS system, a laboratory information management system, to track not reagents and samples, but to track information across all these sites. And then we built around it a simple query portal for the LGRC. Now, when we started building this, we said, well, how do people interact with data today? And if you think about interacting with data, the first thing most people think of is Google, right? You want a nice, clean interface with a box, you type something in, and you get the world's knowledge back, right? Here, we realized that there were more subtle questions that people wanted to ask of data, but there weren't that many. And so a big part of building this portal, we have 6 entry points into this portal, was understanding who the users were, okay? So we built sets of users and we thought about use cases, how people use data. And the first use case was built around people like Sunil, because if Sunil looked at this data that we were analyzing, he would look at how I was analyzing the data, and he would say, John, you're an idiot, let me do it the right way, right? And I would look at his analysis and I'd say, you're an idiot, let me do it my way, right? Every quantitative scientist has their own approach, their own way of analyzing data. Everybody wants to get their hands on the data itself. And so what we did was we built a simple data download portal, but even there, it was focused on trying to make it easy for people to get access to these huge data sets, because nobody wants everything. What you want are separate sets or different sets to ask specific questions. And so we built it like an e-commerce site in a way that what you do is you come in and you have a variety of different metadata that you can use to filter the data. So you can select data types, you can select cohorts that you've defined, and then as you do that, you end up loading up a market basket, a bundle, and then when you've loaded up the bundle with the data you want, you click download, it pulls all the data from the database, it packages it, and delivers it to you. Okay? So very simple, very easy, but giving people who want all the data. On the other hand, we recognize that most people don't want all the data. And so we looked at our clinical and laboratory scientists who were working with us, and when we did, what we recognized was that they wanted something different. They wanted to ask about genes. What is my gene doing in this data set, right? The question you almost always get from somebody who's doing traditional biology. Or what is my pathway doing? And a pathway is just a collection of genes. So we built a gene catalog. And again, it's built around the same sort of idea. Like Google, there's text complete, so you start typing in a gene name, it tries to figure out what you're typing in. And there's all this metadata on the side. There are pathway databases, there are Go terms, and what we do is we go into those data sets, and as you start to type this in, and if you put in AKT1, what happens is all this dynamically updates to tell you the pathways that have AKT1, or the Go terms that contain and to describe AKT1. And so it allows you to drill into collections of genes or gene sets, or functionally annotated gene sets very quickly and very easily. And then once you have those gene sets, our philosophy is anytime you select anything, we should give you something back. If you pick a gene, you're not only going to get just a gene, but what you're going to get is a snapshot of the data that we have on that gene across multiple different platforms. So here we have RNA expression from arrays. This is actually RNA-seq data being shown. There's charm methylation data that we have for this gene. And what it's showing is the distribution across COPD patients, ILD patients, and our normal control patients. Okay? So you get a snapshot on the disease, and already you start to see, well, yeah, for this gene, AKT1, there's actually a difference between our ILD patients and our COPD and control patients. So it allows people to start to generate hypotheses without having to do a lot of analysis. We're giving them hints and laying breadcrumbs so they can start to follow them. But again, we recognize that there are other ways of accessing data. Some people ask questions about locations in the genome. There are a lot of genome browsers that are available. When we started this project, we promised ourselves we would not build a genome browser. So we did. What we did was we looked at all the available genome browsers and weren't very happy with many. So in this case, we took the UC Santa Cruz genome browser, which is probably the most widely used. But if you look at that website and use the genome browser, it's so 1990s in its technology. So what we did was we basically took the data structures, which we really liked, and we embedded it in modern navigation tools. So for a long time, we were calling this genome browser the Franken browser, because it was actually UC Santa Cruz stitched together with the NCBI genome browser and JBrowse, another genome browser. So we took the best of all these open source tools and we put them together and we made something that's really slick. It has multiple windows. So you see a little red box here. That's this window here, and I can have another window, sort of this green looking box, and I can scroll these guys back and forth and align them and put different data sets in them. So we built a genome browser, but ultimately the question we want to ask is a cohort comparison question. And so this was the video you didn't get to see. But what I'm doing here is I'm just looking here at COPD, and I'm stratifying these patients by gender, and I think I'm taking males. I can't read it over there. But I'm taking males, and then I'm looking at their cigarette smoking status. And essentially what I'm doing with this interface is generating pivot tables on the fly, and from those pivot tables I can select a row to represent a cohort, and that's going to be the data that I'm going to analyze. And then once you have cohorts, obviously what you want to do is compare them. So again, a very simple tool, you put in your cohorts, and then we compare them using a choice of methods across every different data type we have. Okay? So whenever you put in a cohort, we contrast them across gene expression, across RNA-seq, across methylation data, and we report something back. And unlike the quantitative scientists who might quibble about what method we use, the truth is most of those methods are pretty close to each other in what they give you at the end of the day. If there's a big signal, you're going to see more or less the same big signal. And so for these very simple analyses, we make a rational choice and we return data back. So in this case, this is looking at differences in gene expression. It's actually showing you the probes on this gene expression array. And one of the other things we could do is look at enrichment for functional classes or pathways that naturally comes out of doing this analysis. Okay? So we've built a lot of tools to make the resources we have available. We're actually replicating that now for a number of different big consortium projects that I'm involved in. In order to do this, we've had to engage corporate partners. We always want to find the best tools. I mentioned that the starting point for a lot of this integrated data warehouse was this grant we got from Oracle. We worked with IDBS as part of that project to create portals. We've worked with Illumina on a project I'm going to mention briefly in a few minutes. We've developed relationships with other companies. And what we're trying to do is bring in the best of breed to really address the computational and laboratory questions that we want to ask of these samples and of these data sets. Okay? You have to enable research beyond your own. So I heard today about the Biostatistics core and how it's a consulting core. We used a very similar model. I was recruited to Stanford, or not to Stanford, I was recruited to Dana-Farber. Dana-Farber, no, don't start rumors. I was recruited to Dana-Farber in 2005. They asked me to put together some kind of plan to support broad-based computational biology, bioinformatics analysis. So it took a long time to really get the plan developed and approved, and part of it was working on a model that would work. But in March of in November of 2009, we launched the Center for Cancer Computational Biology. Mick Carell was actually the first associate director. He came from a consulting background in industry. Yagi Wang, who actually worked up through the ranks, is the current associate director. But the goal of the center was actually very simple. Our mission was to provide broad-based support for the analysis of large-scale data sets. And it was initially omic data. I can tell you we're now dealing with imaging data, we're dealing with text mining data from EMRs, we're dealing with all kinds of data. But it was really to facilitate people's ability to interact with and extract meaningful information from data. We built it around a model we call collaborative consulting. It's a way in which it's an overall philosophy in which what we've really tried to do is to work with people to come in and meet with us and describe their experiments, describe the analysis we want to do. What we do is we sit down after their initial interview, we provide a work plan. That work plan is basically a time and materials quote. We tell them this is what we're going to do, here are the deliverables, here are the checkpoints in the middle, the milestones where you can say go or no go, and here's what it's going to cost. And when we first proposed this chargeback mechanism, everybody hated the idea. People said, well, you should collaborate with us. And we asked them what they mean by collaborate. They mean do the work for free. And then you say, well, how am I going to pay for this? And they say, well, can't you get grants just to analyze data? And you explain, no, you can't really do that. And they say, well, don't you have a student who can do it? And you'd say, all right, can my student be first author on the paper? They look at you and say, well, they're not doing the real work. And then you sort of go back and forth. You eventually realize the best way to do this is to charge people. Also, if you charge people, they place value on it. Because if you don't charge them, you have people who did a crappy experiment, and they come in and they'd say, can you analyze my data? And they say, well, it wouldn't really work. They say, well, do it. And so you do it, and it doesn't work. And they say, well, can you try something else? And it doesn't work. They say, can you try something else? And if every time they say, can you try something else, you say, oh, that'll be $6,000 more. Eventually they say, before we do the next experiment, can you help us figure out how to design it so we can actually get something out of it? And so it's really, I think, improved the quality of science by charging people. We run seminar series we don't charge, we run workshops, and we discovered that if we didn't charge people $25 to come to the workshop, everybody should sign up and nobody would appear. If we charge them $25, the room still sells out, but people actually come. So it's been really useful to assign value to things, because if there's no value, there's no value. So anyway, we use this collaborative consulting model. We also do DNA sequencing, and that's been an interesting part of the history of our center and how we actually did sequencing, how it was going. We provide integrative services of really generating and analyzing large-scale genomic data. But what all these resources have allowed me to do is to actually work on a series of what I think are really interesting research projects. And so I'm going to take you through one of them, and this is actually one centered on ovarian cancer, the starting point for us for discovering hidden subtypes. The real question that we started with was a problem that I was approached by Ursula Matalonis, who is the head of our gynecological oncology project, and she said, look, we're dealing with ovarian cancer. Ovarian cancer is actually a pretty rare type of cancer. It is the fourth largest cancer subtype among women, but it actually has one of the highest death rates. And there are a whole host of reasons why, but one of the bizarre things about ovarian cancer is that all patients with ovarian cancer more or less get the same therapy. And what we know is that a lot of patients initially respond, but then become chemo-resistant and progress to the disease, which eventually kills them. But some patients do respond and do well. And so Ursula said, look, we have a data set. We want to try to identify differences between, we have a sample set. We want to identify differences between responders and non-responders. So we started doing this analysis, and when we got through it, what we recognized was that one of the challenges in looking at clinical samples is that often these clinical phenotypes aren't well defined. And for us, responders and non-responders were defined by a mean time to progression of greater than or less than 6 months. Which, if you think about it, is crazy, because if you have to reschedule your appointment a week later, you can move from being a non-responder to being a responder. Okay? And so that the kind of fuzzy definition didn't work, and we weren't able to really get anything out of this, but we said, look, let's step back. Let's also recognize that in diseases like breast cancer, we have a really well-defined molecular taxonomy. Maybe what we're dealing with is a confounding problem where we have multiple different subtypes and we're treating them all as the same group subtype. So we got engaged in a project. This was actually a collaborative project with people at Illumina. It was an array technology they were developing called Dazzle that I can tell you when we started this project worked extremely well. Right now I would not recommend using it. We've had tremendous problems with it over the last few years. But this is one of their pilot arrays, and it had about 6,000 genes on it. And so we extracted DNA and RNA, and we actually generated RNA expression profiles. And then what we did was we tried to identify subtypes. And we used a technique called ISIS to find what are described as robust bipartitions in the data. I'll talk about that in a minute. And we found a major robust subtype that was associated with the expression of angiogenesis genes. We curated all the published gene expression data to validate the separation and the signature we found. And we found other subtypes in the data that aren't as well supported, but actually are closely aligned with some other thoughts about the genesis and development of ovarian cancer, including a hypothesis that Ron Drakekin, who's one of my colleagues at Dana-Farber, has proposed, in which the source, the cell of origin for ovarian tumors can either be the ovarian surface epithelium or the fallopian tube. And we found support for that in one of the signatures we found. This ISIS method is actually very interesting. And Stefan Bentic, who was the postdoc who really worked with me on this project, had worked with Martin Wingren and his group in Germany. And this is a method that they started to develop. And it's really kind of a clever idea. So if we have a big data set, right, the first papers in breast cancer simply took the data and used hierarchical clustering, and there were strong signals and you saw differences. But the question is, what do you do if there's a more subtle signal or subtle difference between these groups? And so what this method does is it takes the data and it randomly splits the patient groups into 2 sets. Okay? So you have group A and group B. Now what I do is I do a gene-by-gene statistical test across all of those genes. Invariably, I find differences between the subtypes. Okay? So any random separation of data, you're going to find differences. If I separated this room into 2 groups, the people on the left, in some parameter, are going to be statistically significantly different from the people on the right if I measure enough parameters, right? With thousands of genes on these arrays, we always find differences. But if I separated men and women, we'd find a lot of things that are different, right? And so the same idea applies here, that if we have real separations, we should have many genes that have a high degree of significance in their expression profiles. So what we do is do a test on all the genes. We take the top 100 genes, and we use Fisher's rule for combining p-values to get a measure of how good the separation is. And so we eventually do this many, many times. We get reasonable separations. And then we do something like k-means clustering, where we swap samples back and forth to improve the separation between them. Okay? So it's a very simple idea, but what we found was an interesting partition. So I apologize, the colors have changed from blue and yellow to red and blue. But the interesting thing we found is that there's a subgroup over here on the right, it's about 1/3 of the patients, that had a very different expression profile from the group on the left. What drove this was genes involved in angiogenesis. And in fact, when we looked at the survival for these patients, what we saw, and I apologize if it's a little difficult to see here, this yellow line is the angiogenic subtype. And there was a small but statistically significant difference in the overall survival. So we got kind of excited about this. We looked at some microRNA data we had, and in fact, found correlation and corroboration with the microRNA data, that essentially these microRNAs between the different subtypes were switched on or switched off, and that correlated with the expression of their targets in these different subtypes. And in fact, the microRNA data could be used to distinguish them. But more importantly, when we took this data set and we looked at published gene expression data, we actually spent months going through pulling out gene expression data from the public domain for which we had survival information. And the story of this paper was we submitted it, and it was in review for more than a year, and every time we resubmitted it, the story got better and better because there was more publicly available data. And so eventually the TCGA data came out, we took all of that data and included it, and what we saw at the end of the day was, and now you can probably see the line because there are many more patients, but for 1,100 high-grade serous late-stage tumors, we were able to see a separation between these groups. And in fact, if we took all of the data, so 1,600 patients, not selected based on the stage and grade, but and type, we still saw that difference persisting. So we see the statistically significant difference driven by the expression of angiogenesis genes, which again made perfect sense, right? Tumors grow, they need to eat, they try to make blood vessels. If they're getting a lot of food and energy, the tumors do more poorly, or the patients do more poorly. So, for us this was really encouraging. We found this additional subtype, this paper was published. In fact, there's now a clinical trial underway at Dana-Farber, and this is sort of the numerology that made us very enthusiastic about this too. People had been doing clinical trials in which they randomized patients to treatment with standard platinum-based therapy or platinum-based therapy plus an angiogenesis inhibitor. And the results were that about 1/3 of the patients in the treatment arm responded. Okay? Now there was no way to corroborate that 1/3 with the 1/3 that we saw. Nevertheless, it was very suggestive that this might be relevant. And so now there's a clinical trial underway where we're actually collecting samples with profiling gene expression. We want to see if we could predict who would have responded based on these signatures. But my real research is built around this idea of building network models. How do we take genomic data and actually build models that are interesting and informative and tell us about biology, that sharpen the questions we can ask? Okay? Why do we care about networks? Well, the thing we're coming to recognize is that biological processes are not driven by individual genes. And that what we really want to do is think about the genes in their interactions with each other is what drive the phenotypes we observe, what drive the biological processes and systems that we want to understand. So even the earliest analysis of genomic data was really driven by this idea. People were looking for co-expression of genes. But if you really dig into why they were so interested in co-expression, the hypothesis always was that these genes were somehow working together to define the different phenotypes. So these correlations actually suggest some type of network interactions. And so what we wanted to do was develop methods that would allow us to find networks using available genomic data. And a lot of this work goes back to work I did probably in 2004, 2005 with Amira Jabari, who was a student and then briefly a postdoc with me. And it was really work that started thinking about how we infer networks. Now, Sunil asked me, what does my training in physics actually have to do with what we do today? And part of it has really informed the way we think about building models. Okay? In physics, we tend to think about the two sort of grand pillars. There's theory and experiment that supports scientific inquiry. But we also recognize that in the middle, there's something that represents the transition from one to the other. Sometimes the theoretical models were very complex and hard to relate back to the experimental data. So there's this well-respected field of physics called phenomenology. Right? Ultimately, what we want to do is phenomenology in biological systems. We want to develop a theory that describes all the interactions that drive these systems. That theory should be embodied in a model. But actually generating enough data to test the model can be difficult. So what you can do is build a phenomenological model. A model that's empirically driven, that's based on empirical observations and based on data you have, that you realize is consistent from the fundamental theory, with the fundamental theory, but doesn't necessarily have to be derived from it, right? We want to build a model that's consistent with what we understand, but may not capture all the complexities, okay? So the question then suddenly shifts from asking, is this model right, to asking, is this model useful? Does it actually tell us something about the biological system we're studying? Does it sharpen the questions? So we've come to really think about this in great detail. And when I first started writing papers about networks, referees would always write back and say, well, how do you know the model's right? And I realize today that's a crazy, stupid question. That right, when you really ask what they mean by right, means does it look like the picture in my textbook? Okay? If you think about a network of interacting genes or proteins, okay? These guys over here, the little balls represent genes and the lines represent interactions. I've come to recognize that those proteins, those molecules, have no innate intelligence. Right? They don't know what network they're supposed to be in. They bind to what they can bind to. They stick to what they can stick to. And if one of these goes away and somebody else comes in they can stick to, you know what they're going to do? They're going to stick to it. Okay? So these networks, we're actually coming to recognize, can rewire. You can delete interactions, you can add interactions, depending on what genes are expressed at different times, or what the conditions are, or how something like DNA methylation changes. That you can rewire these networks. And it's not necessarily asking, is the model right? The question we're increasingly starting to ask is, how do different models compare to each other? And what can those differences in the models themselves tell us about the nature of disease? So I'm going to describe a method I've been developing with GCU Anna, a faculty member in my department who's a trained mathematician, and Kimberly Glass, who studied physics of complex systems. She's a postdoc working with us, and it's based on the simple observation that gene expression, which is where we still have the most data, is driven by a simple transcriptional process in which genes are activated by transcription factors that bind upstream, recruit a polymerase, and make RNA. A very simple physical model. Now, what we recognize is that in the flow of information from a transcription factor, say, to its downstream target, there are actually two active participants. If this gene is going to be transcribed, the transcription factor has to be present, but this has to be able to respond to it. Okay? So communication has two partners. And what we can do is actually think about modeling that. In communications theory, there's this idea called message passing or affinity propagation, and it really says, if I'm going to talk to Sunil, right, and I'm going to tell him about this method, when he walks out of the room, there's going to be a quiz. And if he takes the quiz and passes with flying colors, we know that I told him about the method and that he paid attention. Okay? But if he walks out of the room and fails, right, then either I wasn't talking about the method, or he was thinking about what he's going to have for dinner tonight, or something much more interesting, or the committee meeting he has to go to in 5 minutes. Okay? So he has to be available to receive the information. Right? And what we'd like to do is actually, given the available data, estimate both these functions. And in fact, we write mathematical functions that relate the two of them and iteratively estimate them if we have enough data. Now the problem is we don't have enough data to do that if we just have a simple model like this. But we have a much more complex model, right? If I give all of you a quiz, and all of you pass except my friend, then we know who's to blame, right? Because I'm obviously telling you about the method, and some people aren't paying attention. So, what we want to do is really use that overall approach to understand what's happening in a network. So what Panda does is actually recognize the method we derived. And I can't remember what Panda is. Kimby came up with this name. It's passing attribute something, something, something. So it's a cute name. Nobody forgets pandas because they're cuddly. But I forget the acronym. So what Panda does is it recognizes the fact that because we have a complete genome sequence and we have motif databases, we know the sequences that transcription factors bind to. So I can map out a network a priori that takes all of the transcription factors and all their downstream targets, which basically spans the entire genome. Okay? So one of the things we do a lot is we make a guess as to the right answer. And here the guess is far too complex. But then what we do is we try to update it. We have protein-protein interaction data, we have gene expression data, and we use that to estimate these two functions, the responsibility for the transcription factor, the availability for its targets, and then we iterate the process. We go back and forth until the solution starts to converge. Okay? We have a paper describing this method. It was just accepted in PLOS One, and it's a method I've gotten really excited about. Okay? So we want to apply this to ovarian cancer. We want to use one data set, so we downloaded the 510 ovarian cancer gene expression profiles from the TCGA data set. We normalized the data. And then we assigned subtypes using a simple Gaussian mixture model. And what we found is what we expected, that about a third of them are the angiogenic subtype, 2/3 are the non-angiogenic subtype. And this actually may be important for understanding response. And then we use Panda to map up the network. Okay, so I apologize, the colors changed. I was remaking the slides, and I abandoned it and just used these with a different color background. But what we have are these two different data sets, one for the angiogenic, and they're always going to be in red, and one for the non-angiogenic in blue. We have motif data, we have protein-protein interaction data, and our goal is to identify networks and then convert them. Okay? So when we do this analysis, right, if you think back to what I was talking about earlier, when I compare these networks, the genes in the networks may or may not change. But what's fundamentally important for us is whether interactions occur or disappear. So in the language of these drawings, these diagrams, what we're looking at is the appearance or disappearance of edges. Now, in this model we fit, they're probabilistic models based on the available data, so we don't say yes or no, but we have thresholds that we can apply. Okay? So what we do is we take the high confidence edges, in this case in the non-angiogenic network and the angiogenic network, and we ask, what can we learn about those interactions? And the interesting thing is those interactions actually touch genes. So you look at the individual genes that they're activating, and what you can see is that the activation, these edges, point to, in many cases, the same genes, but often to different genes. Right? And we're best able to understand and interpret biology based on genes. So even though our analysis is focused on these edges, what we're going to do is focus on these genes. Okay? So if you look at the networks, what you discover is that there are 10 transcription factors that have the largest differences in what they're activating. And this is a phenomenon we see time and again in these network analyses using this panda method. That we're not differentially expressing genes, what we're actually doing. Right? We're not differentially expressing the transcription factors, but we're often switching their targets, and their downstream targets are differentially expressed. And that's actually a really interesting model. So if I look at the transcription factor differential expression between the angiogenic subtype and the non-angiogenic subtype, in most cases, there's not really a big difference. There's some exceptions, but most of these aren't very different. But what we see is there are often very big differences in their downstream targets. Right? So the targets are switching. And in fact, if we look at the available methylation data, the methylation data in many cases supports this switching in the downstream targets. And what's interesting is almost all of these transcription factors, in some way, in some disease, have been associated either with the promotion or inhibition of angiogenesis. Okay? So it's very interesting when you think about this. If you change these genes, their downstream targets change, it does something to angiogenesis. But we're starting to see that it's not necessarily the direct effect of changing the expression of those genes, but altering their downstream targets. Now, when you look at this, what you start to recognize is there are different types of interactions. They're in the angiogenic subtypes, inhibitory, not inhibitory, but excitatory interactions, they're inhibitory interactions. They're interactions where they're excited in one and inhibited in the other subtype, or vice versa. And so what we can start to do is really characterize these different edges in the interactions. And when we do this, we can actually look at the genes that are targeted. And one of the interesting things that we find is, again, with these different interactions, we get patterns of expression associated with gene functional classes for the downstream targets that actually really describe what we see in angiogenesis. So there are a lot of remodeling genes involved in changing the base of the membrane, but also genes involved in blood vessel formation, morphogenesis, its cell adhesion processes we understand to be fundamental to this process of angiogenesis. More importantly, though, when we start to look at these transcription factors, what we see, which we got very excited about, is that a lot of these behave synergistically. And if you start to look at the target genes, there are many cases in the angiogenic subtype where different transcription factors combine with a much greater likelihood than you'd expect to see by chance to regulate these downstream genes or to inhibit these downstream genes. And so what this model's allowed us to do is actually to construct a series of networks that we've used to try to interpret what's happening in angiogenesis. So what we see is that actually HIF1 alpha and HIF2 alpha are interacting with ARNS or S1 to start to promote VEGF production in angiogenesis. And then in fact, there are other transcription factors which bind or don't bind depending on the level of CpG methylation in DNA. And these two things are actually associated with genes that downstream are involved in angiogenesis. So what this has allowed us to do is actually to propose a series of potential therapeutic interactions that we could apply to try to inhibit angiogenesis in this disease. So it's inhibiting these bindings that you see here, promoting other transcription factor pairwise bindings that dimerize, that inhibit their downstream interaction, or decreasing methylation so that transcription factors can't bind. Okay? And so these are all very interesting approaches, and one of the things we found is as we start to go through it and look at the available literature, nobody's actually done these kinds of analyses before. But in ovarian cancer, but in different disease subtypes, they've used different drugs that hint at the kind of patterns we see. And one of the most interesting is this treatment with sorafenib in breast cancer. And what it does is it actually produces the kind of pattern that we'd like to see, promoting or inhibiting angiogenesis in the different subtypes that might actually drive appropriate therapies. So I had some slides in here about network diagrams, and I think they were accidentally hidden. So what I'll do is I accidentally deleted them, which is also possible. So let's imagine I accidentally deleted them, because I've already run over. The last thing I want to say is what you want to do is communicate the mission to the community. So this is actually a website we developed for our Lung Genomics Research Consortium. We had to create a community outreach portal. This was a stimulus grant, and about 6 months in, we're still struggling to generate data. We're starting to get queries from people in the government who wanted to know why all the stimulus money was being wasted. So we hired a science writing team and a web development team to put together something that my mother can understand, right? Realizing that it was probably too high a level to communicate with people in the government, but nevertheless, we want to communicate, the lowest common denominator in society. So we put together this website that actually describes why doing genomic research is so important. And one of the things you recognize is that in sales, you have to really use very attractive people to sell your work. But if you come into this website, you can actually find information about pulmonary disease and why, describing why this project is important. This is actually really good for us because what it allowed us to do is to start to communicate back to patients with pulmonary disease. And we've actually got a lot of positive feedback from them saying, this research is important, I'm glad you're doing it, right? And just educating the public can really help drive patients into target research. So I've gone on a little long. I guess the provost has left. But this is really important for driving patient involvement. Patients really want to be partners in curing disease. Unfortunately, one of the things that we see in biomedical research, and I say this standing in an institution that does this, is we all say we want to cure disease. What we really mean is I want to cure disease. There's a big barrier to sharing data. And for me, getting patients involved has been one of the best things for shaking data loose. And if you hold on to data, your patients can get annoyed with you. If we let them know that sharing data is really important. So for us, this educational outreach has been an important part. Sometimes my friends don't like me that much for doing that. But I really see patient involvement as an important part of breaking the logjam and really driving this forward. Okay? So I've told you about the work we've done. The last thing I want to do is just illustrate with a simple example that genomics is here to stay. In 2010, I got a fellowship to go to Australia for 3 months on this little mini sabbatical. There's actually work on variation that Sunil referred to with Jess Marr and Christine Wells. Christine works at the University of Queensland, then she was at Griffith University, both in Brisbane. Jess grew up in Brisbane, so she went and spent the summer with her family. I took my family, we flew down there. We arrived. Australia is very civilized. For domestic flights, you can actually meet people at the gate. And Christine and Jess both met us. Christine drove us to the house we rented, this little cottage in Arden in Brisbane, and then took me to the car rental place. And we drove to the car rental place. I picked up the car. I drove on the wrong side of the road, panicked, back to the house. And I walked in the door, and if your family works like mine, you can see this playing out even before I describe it. I walked in the door, I was holding the keys in my hand. My wife took one look at me, she took one look at the keys. For the next 3 months, she drove, and I drove the bus. So it was actually really useful, though. The second day, Brisbane has a phenomenal bus system, the best I've seen anywhere in the world. They have subterranean routes, they have elevated routes. And the buses are spotless. On days when there's sporting events, the buses are free. And so I was there, my second day there, I looked up and I saw that this was a good place for doing genomics. There was a sign that said spitting is unacceptable. Bus operators are not equipped with DNA kits. So So genomics is here, quote from William Gibson, the future is here, it's just not widely distributed yet. And last but not least, since I started with a quote from a physicist, I feel I should end with one. So this is from Enrico Fermi. Before I came here, I was confused about this subject. After listening to your lecture, I am still confused. So I'm hoping I've now left you all highly confused. That means I've done my job. So thank you for the kind invitation. I apologize for